Hello, welcome everyone to our next seminar in our seminar series for this year. I'm Carlos Bosques. I'm the CEO at FOSSI. And at FOSSI, we're focusing understanding, translating, and intervention with food allergies. And one of our key questions in research is to address why is there such a rapid increase in food allergies over the last decades. In this seminar series, if you have some of you that have joined, we're particularly interested in what we call Project SAFE, which is really looking to understand the impact of chemicals in the food and the environment and how that may have an impact in food allergies. So over the last few seminars, we've been going through different topics around the areas of what are the chemicals in food and how that may impact allergic diseases. And today we are honored to have Dr. John De La Parra. Dr. John De La Parra is the Director of Global Food Portfolio at the Rockefeller Foundation. And John has been focusing, a lot of his research has been focusing on food crops and medicinal plants and how that impact human health, culture, and the environment. Specifically, John has been directing an effort on the periodic table of food, which he will tell us more about it today. Before we start, I want to remind everyone that we have the Q&A box in your Zoom, and we'll be taking questions at the end of the presentation from John. So with that, John, really excited to have you here. Thank you for your time, the great work that you have been doing for a long time now, and what you're doing at the Rockefeller Foundation is very inspirational to us. As you know, FOSSI is really focusing more on the biology, on why food has an impact, especially in the nervous and the immune system. But another big black box is what are all the chemicals in food, as you know? And so we ask ourselves that question, and how can we understand that to be able to connect that to how food is sensed by the nervous and the immune system. So we thought no one better than you to tell us about that black box and all the information that we don't know about in terms of the chemicals in food. So thank you, John. Great to have you here.
Perfect. Thank you, Carlos, so much, and thanks everyone at FASI and everyone who's here. Really appreciate this opportunity to talk a little bit about the Periodic Table of Food Initiative, which I've been working on for probably over 5 years now. I started my work at MIT and Harvard, where I was a research scientist, and as Carlos said, my interests were in how, in particular, how indigenous people chose food and medicine, and how that choice changed the chemistry of those foods and ultimately resulted in human health impacts. And this was particularly relevant with medicinal plants because there's a lot of interest in how traditional communities use plants to treat certain illnesses. And what we see is that the choice, whether it's the time of year or the part of the plant or the variety or how it was grown, all these things can change the chemistry of the plant and then ultimately change human health outcomes. And that's really part of what the inspiration for some of this work is. So I'll share my screen now and talk for about 15 minutes about what we've been working on during that time. Great. Okay, so the Periodic Table of Food Initiative, as Carl said, I'm at the Rockefeller Foundation, and we've been funding this. We have a couple really great co-funding partners that have helped support this project. But one of the first things that we did was to select a really great secretariat, which is a coordination between the American Heart Association, which is a global research, supports global research in human health, and the Alliance for Biodiversity in CIAP, which is part of the CGIAR, which is a global consortium of agricultural research stations. So what is really unique about this project is we have a combination of world-class human health research and world-class agricultural research coming together for really the first time to look at what's in our food. And food has united these two great institutions to drive a lot of the work at the Periodic Table of Food Initiative. Our vision really started with, as multiple projects that we do at the foundation, with looking at making sure everyone has enough nourishing food to eat. And of course, the Green Revolution, which was about 50, 60 years ago, it started to make sure people had enough food to eat and simply looked at calories, but having enough nourishing food, food that nourishes not just our bodies, but also our culture and the planet are very important. And everyone deserves to know what they eat. They have a right to know what they're eating. And as Carlos said, especially when we're thinking about allergens, we often have a very incomplete picture of what is in our food. Most food composition databases might look at about 150 compounds in our foods, but we know there are thousands and thousands, tens of thousands of compounds in our foods. So we're using that. We're using the collection of those empirical data to really help tell a story about food and ultimately lead to scientific breakthroughs. Um, the periodic table of food itself is a global ecosystem that has really been built to map all of the planet's edible biodiversity over time. And some of the key priorities are focusing on food quality as a key factor. So I mentioned, in the past, quantity has always been the primary, especially for philanthropy. If we look at the investments that have been made, most of it is making sure there's enough calories, which is, of course, important. But this gives us an opportunity to also look at food quality as a key factor, not just in human health, but also in planetary health. The PTFI also maps food quality using a much more comprehensive list of biomolecular data. So something well beyond that 150 that we typically see in food composition databases. And really, we have other, I'll talk too about some of our educational work, which aims to empower stakeholders, people in the places where they are with culturally relevant foods to look at the foods that are important to them. So really, this is about creating a global commons of what is in food. This global commons shifts us well beyond the nutrition label. So most of us look at food, we see this, and this certainly isn't going to tell you very much about things that you might be that people might be allergic to. And it certainly doesn't tell you all the detail of what types of fats, what types of carbohydrates, and other components that are very important to an integrative food systems approach, where we know, as I mentioned in my early work, looking at how indigenous communities made selections for medicinal plants, it would change the chemistry of the plant and ultimately change human health impacts. When we take a food systems approach, we can actually look at the impact of multiple factors, including food choice, from culinary diversity to soil health to ecosystem biodiversity. All of these things we can now tie to something around food composition. I skipped a couple slides. Hold on. One second. So to do that, we built this global ecosystem. So as you can see on this map, we have centers of excellence on every continent except for Antarctica. We have partner labs all around the world, and all of them are helping us co-create what do people want to know about food, how can they measure it, and also to develop standardized methodologies that we can analyze foods and actually cross-compare them to do real science for the first time. And the way that we're doing that is through our foundational resources. So the first part is tools. So as I mentioned, developing standardized tools that the whole world can use. As things were in the past, people would measure all types of components of food, but they often weren't cross-comparable. Data, so having just core data on foundational foods is very important, and I'll walk through a little bit of preliminary data that we have. And then training, as I mentioned, making sure that everyone in the world is able to use these tools, understand how to use food omics, or the comprehensive collection of components that are in our food, to ask and answer the questions that are unique to them in their areas of the world. So the first question, tools, it really is to answer what is in food, and to be able to do that and able to scale those data and to have empirical data that can be scaled, we need standardization of those tools. So we invested a lot of time and effort to develop standardized methodologies in these multiple omics platforms. This has been described by actually your speaker that will be next month, Laszlo Barabasi, who we worked with early on this project, as the dark matter of food. And we see that there's a small sliver of what we actually know is in our food, and the rest of it is yet to be fully elucidated in most of the foods that we have. And this can be particularly important for unknown allergens. So when we look at what is in our food, we can think about the endogenous, or the molecules, so the molecules that are in the foods themselves as it grows. Let's say in this tomato, it's all the components that grew inside the tomato, and then there could be allergens in there, of course, and then the exogenous molecules, so things that might land on the outside of the tomato naturally, or products of fermentation are a good example, so any processing artifact. And then there's synthetic compounds, artifacts of agricultural interventions. So we really need to look at all of these different components to have a full picture of what is in our food, and the PTFI allows us to look at all of these things. When we do that, we need to think about, well, how are we going to do it in a way that anyone anywhere can measure their food, and we can compare apples to apples, literally. Well, early on, we did some work to look at what is the actual need for standardization, and we took the same exact apple sample, and what we found is that, and we sent it to 3 different labs that were very competent in their field, that were known to deliver great data. That same apple sample, those labs did a very good job internally of reproducing their data, so they could reproduce pretty much the same exact data time and time again with the same sample. But the differences between those labs were vast. So of the 927 named compounds reported among these 3 labs, only 14 of the identified compounds were agreed upon. So we see this. So this really clearly and starkly shows the need for a standardized technology in omics, and omics meaning the broad idea of looking at every compound that's in our food. And what that leads to is, now that we have these tools, up until now, we've lacked the tools to understand how the interactions of genes, so the kind of basic programming of how our food becomes from a seed to a plant, impacts chemistry. But now we can actually look at what we call the metadata, so all the things that impact what a food will be, and then the data themselves, all the components, all the actual chemistry of those foods, and we can start to tie those all together. And I'll talk a bit about the different demonstration products that we have. So, okay, so that was the tools. So that's the need for standardization and the different tools that we develop for standardization all around the world. Now, the data themselves are really powerful. So what we've done is we've actually analyzed thousands of foods, but of those thousands of foods, 500 of them we consider are kind of foundational foods, and we're expanding on these every day. But so far we've done 500 foods from 200 different species, and these foods have pretty much every analysis that can be done in a standardized way on them. So everything from proteins to lipids to carbohydrates, the kind of major classes of components, but also multiple types of metabolomics. So those are all the specialized metabolites that a plant makes that make it very unique, that allow protection in nature and flavor and all those other things. We also collect lots of metadata. So when you have an accession into the database, it's not just an apple, but it's an apple that was grown in a specific place at a specific time of year, maybe was processed in a specific way. And all of that allows you to start describing how those different pieces of metadata impact the chemistry. So maybe growing it this way has a different chemistry than growing it another way. We've also developed access and benefit sharing protocols, because a lot of this work is done in the global south and parts of the world that are developing, and we need to make sure that when folks share data about their food, that there are ways that they can benefit, and the data don't just get utilized by others without attribution. So this has allowed us, even with just these 500 foods, we have the deepest and most comprehensive database of food composition in the world. No database has this amount of data on this many foods created in a standardized way, and it really allows us to democratize not just the platforms but the data themselves, so people can have access to this. They can use it, they look at it, they can compare all different types of foods to each other, and the data is increasing every day. Here's a little sneak preview of some data that we're just starting to analyze, and I'll say we officially launched in April of this year. We still are gathering data, and publications are forthcoming in the next year or two. But this is some data from an experiment that we did over the past couple of years to look at regeneratively grown apples versus conventionally grown apples. And what you see here, a regenerative agriculture score, this is 0 through 8, and 8 is the highest, the most regenerative. And regenerative agriculture is kind of like organic agriculture, so it's done in a more environmentally sound way. 8 is doing multiple practices that make it the practice much more regenerative. 0 is fully industrial, conventional agriculture. And what you see is that when we look at the composition of these foods, there's a very clear clustering of data that shows a difference between the foods that are grown regeneratively versus purely conventionally. And then we're just starting to tease out like what exactly this means and how we can parse it a little bit more. And here's a much more complex picture of just apples. So it's very difficult to see on this, but what you see at the top are different varieties of apples. So you have the Envy apple, the Fuji apple, the Gala apple, Honeycrisp, and then for each one of those, you have multiple places in the world where they were grown. So each of these columns represents a place in the world that a particular variety was grown. And then across is different minerals that are in those apples. So this is purposefully kind of like a bit overwhelming because what you see here is that an apple is not an apple is not an apple. Not all apples are the same, depending on where they grow, the variety, and the production method. So there's also in here the regenerative versus conventional. The apple has different components in it, and this could be true for all types of things. This is just looking at the minerals. There's additional apple varieties. Training. So that was the third component I mentioned. So now we have all this data. Who cares? How do you use it? We have data on food already, and people aren't necessarily using it in all the ways that you can. So it's important that we have developed these capacity strengthening projects. One of them is called Food EDU, which is online modules that teach food omics. So how and why do you collect all these data and what do you do with them? We have multiple modules that we built over the past year and a half. And also protocols for how does a laboratory take in these samples? How do they collect the data? How do they interpret the data? So that this is even more democratized. So any lab in the world can look at these protocols, can watch videos and get instruction on how to do it. And then through the food omics and society work, actually look at how then you ask and answer questions around food chemistry and diversity in your own laboratory. And then the Good Food Fellows, which so far we have about 40 to 50 fellows, which are scientists at various levels, mostly graduate students, postdocs in the Global South that we fund to ask and answer these questions around food omics, to use PTFI tools for culturally and locally relevant questions that are relevant to their communities, and then use the PTFI tools. We give them access to that, we convene them, and then we develop this community of people that can use the tools and ask questions that are relevant to them. So and then we also have demonstration projects that those scholars are doing, as well as demonstration projects that we fund directly that help us answer a number of questions. Not just what is in the food we eat, but as I mentioned, how does it vary based on how the food is grown or the variety or any number of things? And how does that ultimately impact people and planet? Here's a bit of kind of the vision for the future. We have phase one, which is research, and that's really we're doing that now, and we've also developed the tools so that we can actually ask and answer these questions. What is in food? What is in food that is important? And now we're working to get that to practitioners, developing data tools for decision making. And we have a number of projects that are utilizing AI and machine learning to integrate those data and develop things like scores so we can understand this complexity in something that the human brain could actually wrap itself around. And ultimately, we want to move into policy so that these have impacts wider into society. So that is my time. If you'd like to check out in more detail the Periodic Table of Food Initiative, you can go to www.foodperiodictable.org. The tool itself is at ptfi.versobio.com. And of course, feel free to reach out to me if you'd like more information, and I'll pause there.
Thank you so much, John. I really appreciate this is a very interesting topic to us. And we're gonna go to Q&As before we jump into Q&As. Just as a quick answer, as I know people always ask me, how can you help with FOSSI? Donate, host events, follow us on social media. You can actually scan this QR code to connect with us. So hopefully you guys can continue to join us on this. And as a reminder, and as John mentioned, our next presentation is going to be by Alberto Barabasi from Northeastern University, who will talk more about the universe of food chemicals. And so with that, I'm going to go into Q&As, and I'll actually just get this started. John, from a food allergy perspective in our communities, and especially thinking about our audience that are non-scientists, we always think about food allergies. We think about the allergen, like the peanut, etc. FOSSI is very, in this project SAVE that we talk about, we're very interested in not the allergens. We're interested in all the other chemicals that are in food that we typically don't pay attention to, because some of our research is now opening our eyes of how those chemicals can also activate the nervous and the immune system, therefore making us more prone to develop food allergies. So kind of as an adjuvant effect, things that exacerbate that. And so in terms of, I think one of your areas, right, is implication of food in human health. I'm wondering if from your current observations in plants or chemicals in plants, or in any food, has there been something surprising that you guys are seeing these new molecules that people haven't seen before in an apple or in any kind of food that you said, wow, that was surprising? And you show us a variability, but something surprising that makes you feel like, oh, okay, that could have
Yeah,
an impact in human health just beyond nutrition.
absolutely. Yeah, no, so thanks for the question. Yeah, and yes, I have. I because some of those data are not published, maybe I won't be too specific, but actually it's we are seeing heavy metals in things that you wouldn't expect. So yeah, things that aren't typically reported as having heavy metals in them. Uh, and then some things that you might expect. So a lot of like sea vegetables, like seaweeds and things, have sometimes alarming levels of heavy metals in them. At least from, and again, these data that we're generating are research data, so like they're meant to be analyzed and looked at. They're not they're not like what the FDA might report. So the data aren't as I don't want to say rigorous, but you know, when the FDA publishes like their food composition database, it has to do multiple levels of review, and it's they through very specifically approved methodologies. So we present these data as research data only for the community to use. But yes, but back to the question, I yes, I think that mineral levels and heavy metal levels are not something that I was really thinking about when I started doing this, but we do see them at higher levels than we'd like, and we see variation, especially based on where they're grown. So you saw that actually just that figure. It was on that figure of the one that was very colorful, had all the different apples grown. You could see like a specific variety of apple. One of them grown in a specific place had cobalt in it at you know interesting levels, let's say, and the other ones didn't. Um, and those are exogenous, right? So it's, you know, those are coming from the plant isn't, you know, uh, making those compounds as metabolites. It's being absorbed into the plant. So yeah, I would say that's probably one, but there's lots of things. I mean, and then, you know, there's things that we don't know what we don't know too, right? Like there's the unknown unknowns. So there's multiple metabolites in food that are, you know, we have a structure or we have, you know, an idea of what those compounds are. Maybe we don't even have a name for it yet. So there's so many things in our foods that we don't even know.
That is so interesting, John. And those are some of the things that we're very interested in, right? Because we're now finding new biologies and new biology associated with allergies that we didn't know before. And we always think, hey, like you said, an apple is not an apple, a peanut is not a peanut.
Hmm.
There's so many things that in the chemicals in the food that we consume that we need to understand. Actually, associated with that, there's a question here that I think may be kind of a hybrid for both of us. But do we think that food allergies develop because of a lack of availability of some nutrients or chemicals in a plant? Does food help us with development of tolerance? So I don't know how you want to,
I
if you,
think that's outside of my expertise. But yeah, I'm not a medical doctor. So maybe,
no,
yeah,
this is interesting. I would say great question from our side, from the food allergy side. Great question. It's a kind of question that we at FOSSI are trying to answer. And I think again, looking at being able to crack some of this biology that's important for food allergies, and then complementing and probably partnering with John that have a better understanding of the chemicals in food and what do they mean for a reaction to our body, especially reactions that are not the typical reactions of toxicity that people know about, but sensitization, etc., would allow us to answer your question. So yes,
yeah,
we'll be interested in answering that question, and hopefully partnering with people like John, we can tackle the chemistry and the biology of it. Um, a few other questions here, John, for you. We have 3 more minutes. How do you standardize for the evolution of live food, like an apple that changes so much as it gets ripe and it oxidizes over a few days, and the chemistry change dependently
mhm.
of the growth conditions?
Yeah, no, that is one of the fundamental questions of analytical chemistry. So you've touched right on any graduate student that goes to study analytical chemistry. One of the first things that you think about is how does the analysis itself impact what you're measuring? Um, so it's a great question, and what we have the simplest answer I think is that we have to just choose what we're going to accept in terms of inaccuracy, and then you do it the same way every time. So whenever you measure anything through, we use these complex machines called mass spectrometers. When I look at an apple, you have to cut up the apple. You have to freeze dry it essentially. So you very cold temperature, you freeze it, you crush it up. So the food's being changed in all these ways, and then it has to be extracted. So essentially, you're making like a tea out of it. So you're pulling it out with either ethanol or methanol or water, and then you're injecting it through a machine. And there's all types of things that may happen in that process. It's about doing it the same time every way, so you can compare a bunch of apples to each other or apples to oranges, and you know you at least did it the same way every time. So and we do have protocols. So like, yes, an apple will get, so and we have, when we collect, remember I mentioned this idea of metadata. So you can record the ripeness of the banana or the apple, because of course we just know by taste that a green banana tastes different than a very ripe brown yellow banana. So you may record, okay, this is actually we take pictures of everything. This is one thing. So you take a picture of the food and maybe you record some metadata about that particular food, and then you have all that information, so it's tagged to the data themselves. And then, this could warrant a study in itself. You've actually described a study. So you could take, 20 different apple samples, cut them and wait 3 days, 2 days, 1 day, and then see how the chemistry changes. Maybe it doesn't change much in what the instrument can measure. Maybe it does. But yeah, so basically we have standardized ways of doing it. Any way you analyze the food is going to introduce some kind of compromise.
Great. John, I would leave us with just one thought of, for food allergies, we know allergens, peanuts, for example, that come from in some countries don't have the same reaction as peanut in other countries, or people don't have the reaction to them. And sometimes we think about the allergen, but other things in those peanuts, for example, will be really important to understand to our audience, especially people living with food allergies. And beyond that, not the allergen, but the other chemicals in food that can activate some mechanisms that can serve to kind of activate our immune systems. And so I want to thank you, John, because these questions that we're having and coming from the biology, really working with people like you that are really cracking the nut in terms of the chemistry, would be really good to work together and try to answer these questions. So hopefully our audience would have good answers to the questions we couldn't answer today, but great questions from everyone. John, thank you for everything you do. Great work, and thank you for being connected to FOSSI. Really appreciate it.
Perfect. Thank you, and thank you for all the work you're doing. Thanks, everyone.
Everyone, bye-bye.